I was a postdoc at University of California, Berkeley. I'm actually currently in Canada, so it's great to see that I'm the second representative from Canada. I'm at the University of British Columbia right now as assistant professor, and I just want to really thank the organizers for giving me this opportunity to talk about something that I think it's really interesting and concerning in the field of genetics. So I'm going to actually start off giving a little bit of a genetics 101 in my favorite organism, which is the fruit fly, Drosophila melanogaster. And in the 101, I'm going to show you some phenotypes we see in fruit flies. So here on top, we have, can you see my cursor?
Yeah. Yes.
Okay, so here on the top, we have what's the wild type phenotype. So the typical phenotype you see from a wild-caught fruit fly. They have red eyes, they have sort of brownish body, and their wings are smooth and they have veins on them. But in the course of almost over a century of genetics research, we have generated a lot of different mutant phenotypes by mutating genes. And some of the common ones we see is the conversion of the red eye into the white eye. Now the body becomes yellow, and now you get these extra hairs on the wings. And geneticists often aren't the most creative people when naming, so they call these different mutants white for the white eye phenotype. So gene names are named after the mutant phenotype. So the white eye mutation causes a white mutant, the yellow mutation causes a yellow body fly, and here the hairy wing mutation due to hairy wing gene causes hairy wings, right? So but what's really interesting people saw was that there are these set of mutations that can be rescued or suppressed by another type of mutation. So when people put mutants, for example, the white mutant, which creates a white eye, with this other mutation that people call suppressor mutation, they actually restore back to a red eye. So they return to the wild type phenotype, and it's the same for yellow. It goes back to the brownish color, and you lose the hairy wing phenotype, mutant phenotype, when you have this additional mutation in it. So these are known as suppressor mutations, and just the name of one such mutation or gene causing this type of mutation is suppressor hairy wing, because it suppresses the hairy wing mutation phenotype. So this is really puzzling because we have these sets of mutations that you can actually suppress the mutant phenotype of. And in the 80s, a set of 2 papers actually came out that were very impactful in identifying what's the cause of these type of mutations, these type of suppressible mutations. And these 2 papers came out around the same time in 1983. One came out in the by a US group in the PNAS, Proceedings of the National Academy of Sciences of the USA. The other one, which came out in the former USSR, came out in the Proceedings of the National Academy of the USSR. So I bet there's sort of some kind of vying for being first. And in these 2 papers, what they realized, and really with almost identical experiments, is that all these genes that cause the different mutation, they have this extra bit of DNA inserted in front of them. And what they realized was that these extra bit of DNA, they're all the same type of DNA, and they're what's known as the transposable elements, what are known as jumping genes, and we'll have a little bit of explanation for what jumping genes are very quickly. And because of these extra DNA, they actually causes a mutant expression of the downstream genes. So you basically end up creating a mutant yellow phenotype, creating the yellow body, or mutant white phenotype, creating the white eye, right? So when you identify these new extra genes, it's common in genetics to give it a name. The US group called it Gypsy, named after the migratory nature of the Romani people. They're people that the people that have migrated into Europe in the early 10th century and are basically all found across Europe. The Russian group or USSR group called it MDG4, which stands for mobile dispersed gene 4, because there were 3 other such transposable elements, 1, 2, 3 prior to this. And so this is where we're getting the crux of the issue here, is that in genetics, people name things, and often many different labs come up with this different names for the same things. It was because people work on just similar projects. And what actually ends up winning out tends to be the gene that has a more evocative name. So these kind of like abbreviations, your kind of boring abbreviation, tend to end up losing out to something more evocative. And this is, so in this case, the name Gypsy won out, and probably also because no one read this paper that's not in English, unfortunately. And this is actually kind of a cherished tradition in genetics to have these funny, tongue-in-cheek or evocative name. So here are some examples, some of which are my personal favorites. So there's a family of genes called the WTF gene. This is sort of a backronym. They selected it just to have a funny gene name. Another one is called Indy, which is a mutation that causes flies to be really long-lived. So the Indy stands for I'm not dead yet. And another one, which was identifying flies, that they called hedgehog, because in development, when you have a mutation in the hedgehog gene, the embryos have these spikes on the back, making them look kind of like a hedgehog. And this gene was also found in humans, and the humans took it and called it sonic hedgehog. So a little bit about what transposable elements are. So these are shellfish genes, or also known as parasitic DNA. They were discovered by the venerable Barbara McClintock, who's a personal hero of mine, and she basically found these genes move around the genome, and that's what causes the different coloration in corn when you have purple and yellow corn. And she subsequently won the Nobel Prize in the 1980s. So just a little description of what transposable elements are, or TEs. So these are genes that can move around the genome, and their sole purpose actually is to replicate themselves. And this happens either through a cut and paste or a copy and paste mechanism. But the problem is that when they make these different copies of themselves and when they move around, they're actually deleterious for the genome. They can make breaks in the DNA, or they can jump nearby or into genes and disrupt the gene function, which is the case when we saw the white, yellow, and hairy wing mutations, they causing these mutations. So because of that, they're often known as parasitic. They cause problems. They also decreases host fitness. But this is why I'm super interested in them, is even if they're all deleterious, they also contribute to a major part of almost all eukaryotic genomes. They're about 50% of the genome in humans and mammals. And in my favorite organism, fruit flies, they're about 25%. In some plants, the common lab species, Arabidopsis thaliana, they're about 20%, but there are species where they're up to 80, 90% of the genome. So coming back to the Gypsy element, so since its discovery in flies, people actually found a lot of relatives of this transposable element. It's found in plants, it's found in fungi, it's found in mammals. And the word Gypsy really came to stand for this entire family of elements that is found basically in all sorts of species. And in some plant species, there are thousands and thousands of copies of these all littered throughout the genome. And unfortunately, because of the name, all of a sudden we are associating all these things we say about transposable elements, that they're selfish, they're parasitic, they're detrimental, deleterious, ubiquitous, or often they're even called junk DNA, to a group of people, right? So a group of people that are often subjugated and oppressed. So I can't really describe this as well as one person I talked to about this, who is a person of Romani Heritage, and I'm just going to quote her here. So she said, "It's a name that many of us didn't choose for ourselves, one that has been used against us to vilify and demean our characters and culture and ethnicity without any second impression, because people only take it at name value. To have a gene reflect all of our worst stereotypes, selfish, detrimental to others around it, etc., doesn't sit right with me and probably a lot of others." So to make things even worse, it turns out that Gypsy superfamily not only encapsulate these types of genes, they also encapsulate a group of retroviruses known as Verde. And for me and many of my colleagues, this is very obviously problematic. And for us, this name really needs to go. And when we started talking to people about this in the genetics and transposable element communities, we got a lot of support, but we also got a lot of pushback. And here I just actually thought to start documenting some of the very common questions we get, and I'm going to go over them. So the very common one is whether or not the word gypsy is a bad word, it's a slur. And the answer is, it sort of depends on who you ask, but for many, it is considered a derogatory term that's used to refer to the Romani people. It's used like Del mentioned as a stereotype for them. And you say that to people, a lot of them will end up telling you that, well, I don't think it's a bad word. I see it get used all the time. So I'm just gonna put down what Amnesty International said. In most languages, gypsy is considered insulting and is directed by Roma organizations. And of course, this is the same message shared by many Roma advocacy groups. And of course, we've got to go with the utmost authority and academic discussions. Wikipedia, here's Wikipedia saying it's considered pejorative. Perhaps the best illustration of the stereotypes and negative connotation the word has is how it's used as a verb. And it's very unfortunately exemplified in the title of this book, right? So another common question is we hear is like, there are a lot of people that do embrace this word, so why is it bad, right? And actually a very common example we get pointed to is this band, the Gypsy King. The Gypsy Kings, they're a very popular band. Another example we one have to see almost on a daily basis is this restaurant that's found near the Berkeley campus that serves very affordable Italian food to the students. But for us, the use of this word, right, to describe transposable element was never actually consulted with groups of people who might be trying to reclaim this word or who are proud of this word. And if anything, the association of the word to TEs or selfish and parasitic DNA actually makes it harder to shed it of the stereotypes and prejudices associated with the word. And this really brings up a bigger point, right? Which is that even if the word gypsy is not considered slur and it's not a pejorative, we are still associating a TE, which has a lot of negative effects to a group of people, and that no gene or TE should be named after a group of people. Okay, so one major problem we see with using this name is that the Roma is actually severely underrepresented in higher education. In a survey of higher education in the UK by the Higher Education Policy Institute, they actually found that only 6.3% of Romas have access to higher education. This is in contrast to 40% of people in the general from the general population. So this is really the crux of the issue, is that by using this word, it actually acts as a barrier to entry, right? When we teach genetics, in genetics in undergrad, one of the first classes people take in genetics, they will start seeing transposable elements because they're very important for students to learn. And then when they see this, they will start seeing the word gypsy, and they will start seeing all these negative connotations we use that are associated with transposable elements. And if a single person of Romani heritage sits in that class and hear this, and if a single one of them gets disparaged, I feel like it's a real injustice to what science ought to be, which is that it should be equitable and it should be accessible to everyone. So one question we commonly get, and this goes back to this cherished tradition in genetics, that are we trying to remove sort of the fun in genetics that we have had for a long time, which is to give funny tongue-in-cheek names to genes, and we're sort of robbing the fun of everyone, right? So there are all these examples that we've shown before. And the answer is no, right? We can be fun, we can be cheeky, we can be whimsical without using names that might perpetuate stereotypes or ones that might disparage others, right? So, and in fact, this is not the first time something like this has happened, again, in fruit fly genetics. So in the 70s, there was this gene discovered that causes male fruit flies to be unable to recognize female and males. And so when you have this mutation, the male fruit flies will court both male fruit flies as well as female fruit flies, and you actually get this really funny interactions where you have male fruit flies all lined up trying to court with each other, even in the ring. So here is one fly that's extending its wing as its courtship song. And the person who discovered this gene, again, in a tongue-in-cheek fashion, called it fruity because fruity describes gay people's. And very quickly, someone else, Jeff Hall, actually identified this issue and talked to the initial discoverer of the gene and renamed the gene to fruitless because the courtship is now fruitless, right? So this is not the first time something like this has happened in genetics, but the problem with Gypsy gene is that it's no longer just one gene, it's now a massive family of genes, and it's no longer just in fruit flies. It now affects a lot of other organisms, and it's even encapsulating a complete different discipline, which is virology. So lucky for us, when we were considering and discussing this issue, the Entomological Society of America also started thinking about the same issue, whether there are inappropriate names being used for naming insects. And one of the first names they identified as being the most problematic was the gypsy moth, which is that has been decimating trees and forests in the northeast of North America. So they started the Better Common Names project to basically start changing the names of the gypsy moth, as well as a lot of other insects that have names that are associated to groups of people or names are associated to a country. So, and luckily for us is that they reached out to us when we were doing this to basically tell us their progress of how they decided to go about changing this. And because in their case, they would have an association that's governing the name of insects, the association can make a lot of executive decisions about how to go about changing this. So then they went through a voting process, or at first they had nomination of name and then subsequent voting. And then after voting, the gypsy moth name has been changed to the spongy moth because they're spongy looking and they're squishy. So unlike what the ESA did, or we have a slightly different problem here, is that the genetic community, at least in genetics and model organisms, the community doesn't have the same organizing body. The closest one is the Genetic Society of America, but the GSA actually doesn't really do anything to control gene names. In the human world, they have their own governing body, but the governing body doesn't have any oversight with what happens in other model organisms like fruit flies and Arabidopsis. So all these genetics organisms are commonly used. So in our case, we basically had to do a bottoms-up approach or a more community-driven approach. And so we actually thought this is a very good opportunity to set a precedence to how to go about changing, in our case, Gypsy, but also potentially other problematic gene names. So here's the roadmap me and my colleagues came up with, which is first to identify the problematic gene name and to identify what the problem is, and to contact as a form of respect the researchers who came up with the name, and then to reach out to the harm communities, discuss with the scientific communities who are actually working on this gene. And in our case, it's a very big community because the transposable element community is very big. It's not just flies, there's a lot of plant, fungi, and mammals, mammalian scientists working on this, and to collect feedback from them and to get a sense of what they think. So in our case, we indeed discussed with the original authors. This is the paper that coined Gypsy, and we recruited a Romani scholar in our effort, Dr. Ethel Brooks from Rutgers University, and we contacted Romani advocacy groups, And one of the best resource we had was to use Reddit. There was a board dedicated to Roma individuals, and we actually went on there to discuss these issues with them. And then we had open discussions on Twitter and on Slack with the scientific community, the transposable element community, to discuss this issue and to get feedback from them. And in our case, we didn't actually have to go through a new name because there was actually a name chosen all the way back, and a lot of people just suggest that we reinstate that other name, MDG4. And so we're at this step, and after all this, the hope is that we can then start contacting, when we have enough inertia and a big critical mass, we can start contacting journals and repositories to actually reinforce these name changes. So as editors, as reviewers, when we see people refer to Gypsy, we will recommend that they change it to MDG4. And just at least for a little while, it will be MDG4 and with a parentheses formerly known as Gypsy. So in the course of sort of discussing this with the Roma communities as well as the scientific communities, we actually came up with a couple of things we really had a hard time thinking through, and we don't have the right answer. And this is just some of the things I think I'm sharing to everyone to really get a sense of kind of issues that I don't know what the right answers to. I don't even know if there is a right answer. And the first is whether there is a right balance between scientific continuity versus reparation to the harmed community, right? So in our case, a lot of people worry that we are creating confusion by now changing the name to such a commonly used genetic element, right? But then the issue is if you don't change it, then it's a problem like we discussed. And then, but how should we go about changing it? Some people suggest that we should keep GY because the short form is GY. So in the case of the fruitless gene, they kept the shorthand, which is FRU but in our case, the shorthand for Gypsy was GY. So a lot of people said, can we change it to a name that still keeps GY, right? But others have also argued that by doing that, we're sort of taking the path of convenience without actually fully removing this problematic name, right? So that's a problem. So another issue we had in just talking with the Roma communities is that for them, this is a mess that they didn't want, as the scientific community created, right? So how much can we actually ask them to help us on this issue? And again, I don't know the right answer, and maybe this will depend on the type of communities you interact with. But in our case, there was a sense that it just feels like it's crap we made up and we should handle it ourselves and leave them alone. And lastly, this is a question we get from the community, which is, does it really matter? You're changing a name, but it doesn't actually address any of the issues that the community faces, right? And in our case, we actually got this question a lot from the Roma communities. And for us, what we can do beyond changing this name is to bring awareness to all the issues they're facing, the people are facing when it comes to entering higher education, the barrier entry, and we're working at efforts for the various scientific organizations to basically have more inclusive programs to help with this underrepresentation issue that the Roma communities are facing. Okay, so with that, I would like to wrap up. So I just want to acknowledge all of my collaborators and colleagues. Most of them are Drosophila T people and my friends, and the person who really helped us kind of navigate a lot of the communities is Dr. Ethel Brooks, who we brought on, and she's a professor at Rutgers. And I just also want to thank all the various Roma communities we discussed, as well as Lee Greenwood, who was the liaison for us with the Entomological Society of America, as well as the Transposable Element Slack channel, where we did a lot of discussion, and a bunch of Drosophila or transposable element researchers that we talked to about this issue. And if you want to read more, we have a preprint about discussing this issue on the Open Science Forum, and you can just search FMA 57 on there. And with that, please let me know if you have any questions.
Thank you so much, Kevin. Does anyone in the room have any questions for Kevin? Or anybody online, in fact? Yes.
Can I ask a question? I think,
Sure.
Kevin, just in relation to the methodology for contacting the journals and the repositories, how did you go about doing that, and how responsive were editors?
So we haven't gone to the journals, but we've been doing working with the repositories. So there are repositories basically for genes and in this case transposable elements, basically a database that kind of keeps tracks of all the transposable elements. And it was really just pestering them, emailing them a lot of times. And some of them want to see it published somewhere. So that's what we're working on right now, is to get this work published. But others sort of just sees this as understand the issue, and they're very open to just changing it. And so journal, we discussed it with some editors. We haven't widely distributed this. And partly it's still not full, it's just a preprint right now, which we haven't published, right? But I think a lot of it would just be emailing journals about this. And I think a big part is to get the word out there. And this is why we sort of made a fuss about this on Twitter and also got the preprint out, is for researchers to see this. And for when researchers see this, they will get, when they review papers, they will be able to say, please use these, to stop using this name and use another name, right? So in many ways, I don't think editors is what we're really focusing on. We're focusing on all the scientists who will act as reviewers, who will then reinforce this when they see articles. And just some of the, we have already seen some papers that have adopted MDG4 and with a little parenthesis saying formerly known as Gypsy. So we're sort of, I feel encouraged that this seems to be actually making a difference. But again, once this paper is fully published, I think we can make a more concerted effort in contacting journals.
Fantastic. Any other questions in the room? And any other questions online? In that case, Kevin, thank you so much. Oh, somebody's talking. Sorry.
One question
Yeah.
from Melia.
Oh, okay, let's have a look.
Would you like to come online, or would you like us to read it out? We can read it out.
We can read it out.
Okay, so not really a question so much as a comment, but as a person of Roma heritage, I'm always darkly amused when people point to the Gypsy Kings as, see, it's not a bad word. Yeah, the Gypsy Kings were not Roma. It wasn't their word to use in the first place.
Yeah, no, absolutely.
And I think that's everything from online.
Wonderful. I want to say thank you so much, Kevin, for coming online so far away.
Yeah, so just one thing to add, I would say the restaurant picture of the restaurant I showed, they're not also not that heritage. I'm pretty sure it's just Italian ran restaurant. So, you know, I agree with you. Yeah, there he is. Yeah.
Fantastic. Again, thank you for getting online so far away. We really do appreciate it, and for giving your talk live. Again, thanks to Kevin.